In this last section of Chapter 13, we look at how we can test the effectiveness of antiseptics and disinfectants. So we've gone through and looked at the different types of chemicals that we can use to control microbes. But we need to have a standard of how effective they are. Remember that we want something that's selectively toxic, that is going to harm the microbes and not harm us. And because of that selective toxicity, we have to figure out the exact concentration and the exact exposure time so that we can achieve our goal of killing those microbial cells. Different types of microbes may require different concentrations or different exposure times. And we also have to consider our end goal. Are we content with disinfection, or do we need to achieve sterilization? We can use several different methods to determine the effectiveness of antiseptics and disinfectants. And the best way that we can do this is really by putting those to the test in the field, essentially an in-use test of what they would really be up against in terms of the environmental conditions. Because temperature and pH, the appearance or the presence of dirt or blood or other bodily fluids can all interfere with a chemical to do its job. So we have the phenol coefficient to help us determine effectiveness, but then we also have the disc diffusion, use dilution, and in-use methods that we can use, and we'll take a look at all four of those. So the phenol coefficient was kind of the first method that was used to test the effectiveness. In this method, a chemical agent's effectiveness was compared to that of phenol. And remember that phenol was what was used by Joseph Lister. We don't use phenol anymore because we realize that it can be toxic and it can be a skin irritant. But traditionally, we would compare the effectiveness of these chemicals to phenol. Okay. Two British chemists, Riedel and Walker, actually established the protocol to be able to compare chemicals to that of phenol. And you see that they used two different organisms. They used Staph aureus as their gram-positive, and then Salmonella enterica cerevar typhi, which is the causative agent of typhoid fever, as their gram-negative bacterium. They exposed the bacteria to the particular antimicrobial chemical for 7.5 minutes, and then they calculated what they called the phenol coefficient. A phenol coefficient of 1 means that it has the same effectiveness as phenol. If it's less than 1, then it's less effective than phenol. If it's more than 1, then it would be more effective than phenol. So we had this kind of first method that was used, but it was just with two bacteria. It was, pretty subjective. The organisms, the conditions, the chemicals were just arbitrarily chosen. So it wasn't a true method of assessing complete effectiveness of the different chemicals. Another method is called the disc diffusion method. In the disc diffusion method, this involves putting chemicals on sterile cotton or filter paper discs that are placed on a plate that has been covered with the bacterium that is being tested. And essentially what happens here is the bacterium has been completely spread over the surface of the agar using usually a sterile swab. A loop could also be used, and this creates what we call the bacterial lawn We want as much coverage over the surface of the plate as we can. Then the discs are placed on the plate. The plate is incubated as normal. Then when the plate is removed from the incubator, we measure the zone around the disc where there's no growth. We call that the zone of inhibition. And we use a ruler to measure the length of this zone of inhibition. So in this particular example here, you've got Pseudomonas aeruginosa, you've got Staphylococcus aureus, and you have 4 discs that have been placed there. And you can see this lawn. You can see how the bacteria growth is in different directions because we really want that all over coverage. For Pseudomonas aeruginosa, you can see that for these 3 down here, there was no zone of inhibition. That tells us that this particular chemical is not effective against Pseudomonas. Whereas if we look at Staphylococcus, we see a zone of inhibition around all of the discs. And that tells us that all of those chemicals are capable of killing or inhibiting the growth of Staphylococcus aureus. The larger the zone, the more inhibition that has occurred. Okay. That's what we use in the lab. We use the disc diffusion method in the lab. Now, 2 other tests that can be used are the use dilution test and the in-use test. The use dilution test is used to determine the effectiveness on an inanimate surface. So we're looking at, how is it going to be used like on a tabletop or other fomites. They take a cylinder of stainless steel that's been dipped in their culture that they want to test and then dried. And then they take that cylinder and they dip it in the solutions of the disinfectant at various concentrations for various lengths of time. Then they put that cylinder into a test tube and they incubate that and look at the turbidity, so how much growth is present in that medium. If the target organism has been killed by the disinfectant, then of course there won't be any growth in the tube, and so it will remain clear, it will not be turbid. If it's very turbid, we have a high level of turbidity, then that tells us that that disinfectant is not effective. The American or the Association of Official Agricultural Chemists International has established these protocol standards, and you can read there, they determined that a minimum of 59 of 60 replicates must show no growth to achieve a passing result, and the results must be repeatable. So, if you look at products that you buy, and they say, they kill this 99% of the time, or they kill this, after 10 minutes, or this after a certain concentration, then what they've done is these use dilution tests, and then gathered all of that data so that they could put it on the label of that product to talk about how it works. The last test we have is the in-use test, and this is when we really put it to the test in a real world situation. So we take a actively used solution of disinfectant and determine if it's microbially contaminated. And this in-use test here, you kind of see what's happening. We have used disinfectant. We take 1 milliliter of that and put it into 9 milliliters of sterile broth that has an inactivator. And then we take a sample of that, place 10 drops on the plates, and then incubate, 1 at body temperature, 1 at room temperature. And then we look for the growth of colonies. And if we get colonies, then that tells that it's been contaminated. And really what this does is it allows us to monitor the use of disinfectant. So if we're using the same disinfectant. Are the bacteria growing resistant to that disinfectant? So this is a good test for that. The important thing to remember when we look at any antiseptics or disinfectants is that we have the instructions for use that are on the chemical. And these instructions for use have been studied and tested and studied and tested. And when you're using a particular product, if it says spray on and leave for 5 minutes, then that is the time of exposure that it takes to kill these particular microorganisms. So it's very important that we all follow the directions on the chemicals that we're using to keep our home safe. That we know when we use Lysol wipes or we use bleach or we use these different chemicals, we want to make sure that we use them at the correct concentration and the correct exposure time so that we aren't part of the problem in creating resistant bacteria.